Cell research offers understanding basic mechanism of human development and differentiation and hold the potential for therapies and cure for disease such as diabetes, heart disease, spinal cord injury, Parkinson, Alzheimer's disease. Rapid progress in stem cells have introduced a host of ethical and policy issues. In India, there is no law to regulate the SRC. Stem cell research. Stem cell. ICMR and Department of Biotechnology has issued guidelines for stem cell research on the National Guidelines for Stem Cell Research 2017 to ensure that all researches within human stem cells are conducted in an ethical and scientific recognizable manner. Understanding philosophy is to prevent the commercialization of unproven and stem cell therapy and generation of new knowledge based on the sound scientific rationally while addressing the ethical concern. An important aspect of the guideline is that to clarify the ambiguity over who has legal jurisdiction over the use of stem cells. Silent features. Stem cells and their derivatives are defined as drugs as per the Drugs and Cosmetic Act 1940. Any stem cells used in patients other than that for hematopoietic stem cell reconstruction for approved indication is investigation at present. Guidelines do not apply to research using non-human stem cells and their derivatives. Hematopoietic stem cells, platelet-rich plasma, autologous chondrocyte, osteocyte implantation. Clinical trial using stem cells should be in compliance with the scheduled by Drugs and Cosmetic Act. Good clinical practice GCP guidelines and ICMR ethical guidelines for biomedical research. Mandatory registration of the Institutional Committee for Stem Cell Research. All clinical trial must have a MCA registered postgraduate medical specialist in the subject domain of the trial. Ethical and scientific issues. Health and safety right of the donor are the utmost important. Human participants enrolled for the clinical trials are not able to pay any charges towards procedure, investigation, or hospitalization related to the trial. Identity of the donor shall be kept confidential at all times. Mandatory video consent is necessary. Mandatory screening for 6 major transmittable disease: HIV-1, hepatitis B, hepatitis C, trypanoma pallidum, human T lymphotrophic virus, cytomegalovirus, and other risk factors for genetic disorders. Intellectual property rights of donated material will not vest with the donor but may be shared to be mentioned in informed consent form. If commercialization brings an financial benefit, it may be passed on to donor and community. Permissible research includes establishment of embryonic stem cells and induced pluripotent stem cells. Restricted research involves human pre-implantation embryos processed by in vitro fertilization and intracytoplasmic sperm injection. Somatic stem cell nuclear transfer to derive ESC lines and prohibited research involve human germline gene therapy and reproductive cloning. Genome modification is restricted only to in vitro studies. In vitro studies can be pre-implantation. Human embryos must be carried out within 40 days of fertilization or formation of primitive state whenever is earlier. Uterine implantation, human and animal and manipulated cells with the intent of developing a whole organism is prohibited. Banging of umbilical cord, blood, or ESC induced pluripotent stem cells and is permitted only in a licensed institution. Commercial banging of all other biological material is not permitted. Procurement: if stem cells or tissues have been developed by utilizing IVF method, permission is mandatory. Archival period of stem cell lines and related information is 10 years. For procurement of fetal or placenta tissue, processor must comply with all obligation under the MTP Act 1971. The medical person responsible for patient's care of and the investigator using the fetal material shall not be the same. Consent for fetal tissue donation should be obtained in advance and not just before or at the time of the procedure. If there is a disagreement between the parents, the mother wish shall prevail. Consent for donation of blastocyst for establishment of human ESC lines should be obtained from the donor at least for 24 hours in advance. Donor retain the right to withdraw the consent until the blastocysts are actually used in a cell line derivation. Important of stem cells lines for basic research will not require non-obligation objection certificate, but those required for clinical trial and originating overseas require important clearance. For export of indigenously developed cell lines, clearance must be obtained. Publicity: the advertising and publicity of probable benefit of stem cell therapy through any mode by clinicians are not permitted. Thank you.